A1C? What's the biochemistry? So, say you want to get an idea if a person is able to control their blood sugar levels. So, no matter what they eat, no matter how much sugar they eat, they're able to maintain a healthy level of blood sugar or glucose in their bloodstream. Now, if you were to just take someone and measure how much glucose is in their blood, that wouldn't be a very helpful indicator because it would depend on, say, did they just eat a bunch of Halloween candy? So we need a more useful measurement. We need something that is going to analyze basically over time what's happening in the blood. And a way that we can do this is by measuring this thing called the A1C. This is a form of hemoglobin, so the oxygen carrying protein that our blood cells are chock full of, is glycated. So it has this sugar chain attached to it. Now, the more sugar that is in your blood over time, the more of this glycated hemoglobin is going to appear. So if you measure, and the hemoglobin this is the blood cells, they're going to have a lifetime of several months. And so I think it's 120 days. And so if you were to measure the levels of the glycated hemoglobin, that would tell you about what's been going on over the last few months rather than just what's happening in that instant. Because what's happening in that instant is going to depend on, say, what the person just ate. But what's going to happen over the time course is it's going to depend more on the ability of the person's body to actually regulate those glucose levels. This ability can be compromised if people have a condition like diabetes, where, say, their bodies aren't able to make insulin, so they're not able to make this hormone that's going to tell cells to let in blood sugar, or their cells aren't able to respond to that message. What happens then is that this glucose can build up in the bloodstream. Now, the red blood cells, these actually use a different glucose transporter that doesn't depend on that insulin. So even if people have diabetes, their blood cells can take up a bunch of this glucose, even if the other cells can't. And since you're building up so much glucose in the bloodstream, if you're not able to get it into other cells, what's going to happen is that you're going to get a bunch of glucose flowing into those cells, and those cells are chock full of hemoglobin, because hemoglobin is the protein that's going to allow your blood to transport glucose or transport oxygen throughout the body. And so you're going to have a lot of hemoglobin in these cells, and you're going to have a lot of glucose in these cells. And what's going to happen is you get this non-enzymatic reaction where basically it doesn't need any helpers. This glucose, just the chemical properties of the glucose and the hemoglobin, are going to make it so that they react and that the sugar gets permanently stuck onto that hemoglobin. And because it's permanently stuck, well, now no matter how long that hemoglobin lives for, it's going to have that modification. And so if you measure the amount of glycated hemoglobin, you get a sense about whether the person has been able to control their blood sugar levels. And so here is a little more detail about the biochemistry, where the naming comes from, how we measure it, how it happens, all that sorts of really cool stuff.
First, let's meet our molecular players. So this is hemoglobin. It has these 4 subunits. So it has these 4 individual protein chains. And then it has these heme groups that are going to latch on to oxygen. So you have these A and B chains, and this is showing you adult hemoglobin. There's also fetal hemoglobin. So this HbF. When we talk about HbA1c, that's talking about being the adult form. That's what the A stands for, and it's not talking about the alpha chain. In fact, the modifications that we're going to see when we get that glycation is actually going to be happening on the beta chain. There are different types of the glycation that can form, but the type that is specifically referred to as HbA1c is happening at the N-terminal valine of the protein, of the beta chain. So proteins are made up of these chains of amino acids, and at the one end, the starting end, you have an amine terminus. This N-terminus, you have an amino group. And this is where we're going to have this modification. It's on this amino group of the amino acid valine, which is on the first residue of that beta chain. And we'll get much more into this later. So that's the hemoglobin. And then the other component we're going to talk about is the glucose, because what's going to happen is that we're going to get a glycated product. So with glycation, there are different ways to attach sugar to various molecules. And what we're talking about here, we're going to have a glycoprotein. So we have a little bit of protein, we have a sugar attached to a protein. And in this case, instead of being added enzymatically, so instead of having a specific molecule dedicated to help add that sugar on, what's going to happen is it's just going to happen spontaneously. And the sugar that's going to get added in this specific reaction in the case of HbA1c is going to be glucose. Now, glucose is blood sugar, and it is basically the main building block sugar that we use to make a bunch of other sugars. If you think about table sugars, now that's sucrose, and it's made up of glucose and fructose, and then this gets broken down, and glucose can get converted into a bunch of other forms of sugar and modified versions. We can see glucose can be in this like ring form or this linear form. And so we're going to see later that we're actually going to see some interconversions between the ring and linear form, with the reaction happening in this linear form, but then it's going to like re-ringify. So that is hemoglobin and glucose, and they're going to come together to form this product. And so how this is actually going to happen is it's going to happen through this Schiff base formation and then amidori rearrangement. So those are some kind of like scary sounding words. But basically what's going to happen is that N-terminal valine, the N-terminal, the N has got this amino group. So it's got this NH3 or this NH2 if it gets deprotonated, and then when it gets deprotonated, it has this lone pair of electrons. So much more on chemistry and the various things in other posts, but basically electrons are the way in which atoms, so individual nitrogens, carbons, etc., are able to form bonds to one another. All of these atoms are made up of these smaller subatomic parts. So you have positively charged protons and neutral neutrons hanging out in this dense central nucleus, surrounded by this cloud of negatively charged electrons. And for various reasons, these atoms want a certain amount of electrons, and so they're able to form bonds with one another where basically they share electrons. They share a pair of electrons to form a bond. Two pairs just form a double bond, and this allows them all to get to their ideal number. And they will react in order to do that. One of the main reasons we get reactions is this thing called like nucleophilicity and electrophilicity. So a nucleophile is something that has more electrons or electron density than it wants, and so it's going to seek out something to share it with. And the electrophile is the thing that wants more electrons, and so it's going to be willing to share. When we see that that nitrogen in our valine, that's going to act as a nucleophile. It's going to have this lone pair of electrons that's very nucleophilic. And so it's going to attack the carbon in this carbonyl. So basically oxygen is what we call very electronegative. It's going to be hogging those shared electrons. When you pull the electrons away from this carbon, it makes this carbon partly positive. And this partial positive charge is going to be attractive to this nucleophile. And so what's going to happen is you're going to get that nitrogen attacking this carbonyl, so the C double bond O of glucose. Then what you get is this thing, so it's called the Schiff base, where you have this double bond between the nitrogen and the carbon. And this is reversible. So we actually see Schiff bases a lot in biochemistry. Typically, we're dealing with lysine, because lysine has one of those amino groups on its end, so on its unique part. So the amino acids all have this generic backbone and then this unique side chain. Now the generic backbone has an amino group, which is why we get an amino group in that terminal valine. But when these amino acids link together to form proteins, well, then the amino group, you don't have this free amino group anymore except at the ends. Instead, when they join together, they're using that amino group, and so they kind of like use it up, and so they can't form that reaction here unless they have an amino group in their, I have one of these primary amines in their side chain. And so lysine does, and so it's able to form these Schiff bases. All of the others can only form these bases if they're on the N-terminus. And so since we have a valine on the N-terminus, the valine is able to form these Schiff bases. But other times in biochemistry, we're seeing Schiff bases formed with lysine because that can happen no matter where in the protein that lysine is. And so this is really useful for doing things like removing nitrogens from amino acids, so like transaminations and transferring amino groups, some really cool stuff. And if you want to learn more about that, check out the post on lysine. But today we're talking about this valine. And so we get this Schiff base formation, And as I said, this is reversible, which is what makes it so great for doing things like transferring nitrogen groups from one place to another. But it doesn't make it so great if you want a permanent change. Thankfully, or I guess I don't know if it's really thankfully, because these glycated products can be like harmful, they can build up. You get these things called like advanced glycation end products or AGEs, and then this can cause various problems that I'm not going to get into. But basically, this can form this rearrangement, the spontaneous rearrangement. It's spontaneous again. We're not doing anything. We don't need any enzymes to help it out. It's just going to kind of rearrange to get comfier. When it rearranges, what's going to happen is instead of having that double bond to the nitrogen, which is reversible, we have this double bond shift over to this oxygen to give us this carbonyl. And then it's giving this ketoamine. So a ketone is where we have a C with a double bond O, so this carbonyl group with 2 carbons on either side. And then an amine is where we have a nitrogen attached to hydrogens. And so here we have a keto, this part, amine, this part. And once you have this, then it can recyclize. And in a bit more formal, the layout here is a figure from Wikipedia where you can see this reaction happening. And if you want more on the details of how that reaction happenings, I actually like walk it out in a post that I will link to. So this is the idea of how this reaction can actually form. And since once it forms, it's not reversible, then you're going to get a buildup of these glycated hemoglobins. And these hemoglobins are going to stick around in those blood cells because they're pretty stable. And the blood cells have a lifetime of about 120 days. So we're talking about 4 months or so. So if a person has well-controlled blood sugar, they're going to have lower levels of this because the glucose is going to be able to get into the other cells and it's not going to build up in the bloodstream as much and get into the bloodstream, the blood cells as much, because that's the only place that they can go if they can't get into other places. But if they can get into other places, then they will, and you're going to have lower blood sugar, lower amounts of sugar in those blood cells. And if you have lower amounts of sugar in those blood cells, then these molecules are less likely to bump into each other and less likely to form these interactions, these reactions. And so you're going to have a lower level. In terms of like what is normal, I am not a doctor, and I am not going to pretend to be a doctor, and I'm not going to give out any medical advice. But according to the CDC's website, normal is below 5.7%, prediabetes is 5.7 to 6.4, and 6.5 or above indicates diabetes. And so if you have high A1C levels, then your doctor might do some more tests and that sort of thing. And if you have diabetes, your doctor might be measuring your A1C over time to see if your treatment is working, whether you're well able to control the blood sugar, that sort of thing. In terms of how they do that measurement, there's a variety of different ways. So here is a review article. A lot of these methods rely on some sort of chromatography. I talk a lot about chromatography when I talk about protein purification, where basically you can take these columns filled with these little beads called resin. These beads have different properties that are going to interact differently with different molecules, depending on the properties of those molecules. And this allows you to separate the molecules as they go through. You can have different types of resin to separate them based on different things. So for example, we can have resin that specifically has something that's going to bind to the glycated hemoglobin. And one thing that will bind well to the hemoglobin is this thing called this like boronate. So you get this like boronate polyanion, this like column that has traditionally been used in order to kind of capture the glycated hemoglobin so that you can then measure it. There are also methods that use some sort of ion exchange chromatography. So separating things based on charge, because having that glycation is going to change the charge of the protein. And there are other methods that use things like HPLC, separate them based on like their polarity and things. There are enzymatic methods where you basically cut off that sugar and then you use various detection methods in order to measure how much sugar there was. There are antibody-based methods so antibodies are these little proteins that bind specifically to different things. You can get antibodies, you can get like animals and things to make antibodies against that glycated hemoglobin, and then you can mix those antibodies with the blood and see how much of that hemoglobin was present. So this is from like my post on like blood sugar types or blood types, but a similar thing can be used where you have some sort of agglutination reaction, where if the antibodies find the glycated hemoglobin, they're going to bind, and then they're going to form like clump up. And so this like agglutination reaction. So those are some of the methods that can be used in order to measure this A1C. In terms of the naming, so you have those different subunits of hemoglobin, and then you can have different modifications on those. And HbA1c is specifically referring to the adult form of hemoglobin, so where you have 2 alpha chains and 2 beta chains, where the beta chain has that is glycated at that N-terminal valine. So where you have a glucose attached to that N-terminal valine. So normal adult hemoglobin is mostly this HbA1c, where you have this alpha and beta subunits. There's also HbA2, which has this delta subunit, and then there's fetal hemoglobin, which has this gamma subunit. But like 97% is going to be this HbA. And then about 6% of that HbA is going to be HbA1. And HbA1 is going to be kind of like a mix of different glycation products. And then a fraction of that is going to be the HbA1c. In 1958, Huseman and Meyering, they actually discovered glycated hemoglobin, but they didn't know it was glycated hemoglobin. They were just running agar starch electrophoresis. So basically, we talk about like agarose gel electrophoresis a lot, separate DNA by size. Basically, how it works is you make this mesh out of agarose, and then you use electricity to send the molecules, attract them to one end of the gel after you load them in the other end. And so DNA is negatively charged, so it'll move towards the positive charge, so we can use it to separate DNA by their size, because the longer strands of DNA will get tangled up more in the gel mesh. And so when we turn off the electricity, they'll be higher up in the gel. There are also other forms of electrophoresis. We talk about like SCS page we use for proteins. Here they were using agar starch gel. But basically what they found was that when they took their hemoglobin, it seemed to be, and they ran it through this gel, it seemed to be like made up of multiple things. There was a portion of it that moved faster than the other portion. So the glycation, what's happening is that glycation is actually, because it's replacing that like NH3 group, is going to be reducing the positive charge, and this is going to make it move faster, because remember, the negative charge is what is going to be propelling it through the gel. So we're talking about, when we say reducing the charge, it's reducing the positive charge, so it therefore like making it more negative, and negatively charged is going to move faster through the gel, so you get this faster fraction. And then this other group, Allen et al., they further separated this fast hemoglobin, this like hemoglobin A1, they then separated this further using chromatography. So they actually have chromatography here and here instead of the agar here. But the same idea is where you have differences in the charge, they're going to affect how the proteins are going to separate. And so if you have modifications, they're going to affect where the proteins run, either through the gel or through the column. Allen, Schroeder, and Balog, basically they took the hemoglobin, they separated it using chromatography. So they were separating it based on differences in their charge, and you get this fast moving fraction. So as things are coming off of the column, the things that you can like collect them in fractions. And then the sooner they come off the column, the earlier the fraction number will be. In this case, this A1 fraction was coming off earlier than this A2 fraction. And when they took this A1 fraction, they could further separate it. And when they further separated it, well, now they found that it separated into these different groups. And this A1B is the one that turned out to be, when they further analyzed it, This turned out to be that valine, that modification of the valine, the glycation of the valine of the beta chain that we now know so much about. And then these other peaks corresponded to like other glycation products and things. But so when they talk about the fast moving peak, this would be your A1. And then when you have this, when you further separate it, then you get this A1B. And so that's where the naming comes in. And so it can move faster because when you have that glycation, you're negating the negative charge of the N-terminus. And so it's going to be less charged. It's going to therefore stick less to the column, come out faster. So then you get this fast moving fraction that you can then further separate. So they call this like rechromatography, which I thought was a pretty cool name. But anyway, it's an interesting article you can read on 1958. I like some classic biochemistry. And this is still relevant today, very relevant today, as we have seen in this post. And so just to recap, if you have a lot of blood sugar, high amounts of blood sugar, normally what happens is that if you're healthy, your cells are going to send out insulin, so this hormone that's going to tell the cells in your body, okay, we've got a lot of sugar out there, why don't you go ahead and let it in? In people that have diabetes, they either can't, in type 2, they can't kind of like respond to that message, and in type 1 diabetes, they can't actually make the insulin, and so they can't send out that message. As a result, the glucose is going to build up in the bloodstream. Now, the bloodstream, the cells, your red blood cells don't depend on those glucose receptors in order to let in the sugar. And so what's going to happen is that those blood cells are going to let in sugar, even though the other cells can't. When those blood cells let in sugar, well, now that sugar is going to end up bumping around a bunch with the hemoglobin. So hemoglobin is this protein that carries oxygen through your blood, and so your blood cells have a lot of it. When you have a lot of this hemoglobin and you have a lot of this glucose, now what can happen is that they can interact, and they can interact in this way that's going to modify the hemoglobin. And then you get this HbA1c product, which is a glycation product that can then be measured. And this is going to be a permanent modification. So it's going to tell you about the relative blood sugar levels over the past 2 to 3 months. So it's a really helpful indicator about overall control, long-term control of blood sugar, as opposed to just an instantaneous measure of blood sugar. And again, this is all spontaneous. So basically the concentrations that you have are going to be influencing how much of these products you get. And it doesn't require any sort of enzymatic help. So you don't need any like proteins helping mediate or speed up the reaction. So it's a really helpful tool. And these products can also, though, cause various problems. You get these things called advanced glycation end products, various like this can get even more modified, various things like that that I'm not going to get into. But the main way we think about it is as this kind of tool to be able to get a sense about a person's blood sugar levels. So hope that helped you understand this. And yes, remember, I'm not a doctor, but there's a lot of biochemistry that comes into play in terms of medicine. So if you're pre-med or something like that, don't scoff off on the biochemistry, OChem and stuff like that. It really does come into play, and it's really, really fascinating. So yeah, so hope that helps.